All right, before we get started, does anybody have any questions over what we were doing last time? Does the idea of what a quantitative trait or an additive genetic trait make sense to you? Okay, yeah.
No, I just
Good.
was trying to remember what that one was, but not remember.
That's when you have multiple genes that underlie a single trait. Each of those genes can be either a dominant or recessive allele, but it's the dominant alleles that add to the trait. So what we want to talk to you about now is evolution, because evolution is just change in allele frequencies. So we have to think about allele frequencies in order to think about evolution. So I'm going to take you through a little exercise. So imagine that we have two individuals, a homozygous dominant individual and a homozygous recessive individual. At this point, we have two individuals. Each of these individuals are diploid. They have two chromosomes. Each chromosome bears one of the alleles. We have four alleles total because we have two individuals here. Two of those alleles are the dominant allele, two of them are the recessive. So the frequency of the dominant allele is 50%. The frequency of the recessive allele is 50%. And you're going to see me referring to allele frequencies in their fractional state. And there's a good reason that I do this. So I won't be calculating decimals, because decimals just get complicated in ways that fractions don't. So if you're not fluid with fractions, hopefully you will be somewhat. What's the first thing that you do when you're doing a cross? Find the gametes. Big A gamete from this individual, little a gamete from this individual. You will combine these together to produce a new generation of individuals that are heterozygous. That's what's going on here. For some reason, my clicker is not working. Oh, because I don't have it turned on, that's why. So it doesn't mean that these two individuals only have one offspring. They could have 100 offspring, but all of them are going to be this genotype. All of them are going to have a big A allele and a little a allele, so you can also think of it as being one individual. There's two alleles because it's diploid. One of these is the big A allele, one of these is the little a allele, and so we're still at frequencies for P and Q of 1/2. Now we're going to let these guys interbreed. These individuals can each make two different gamete types. And we can put them into a Punnett square, and it's the easiest Punnett square on the planet to operate. And Dr. Jed taught you to think about these in terms of genotype frequencies. So we could have a 1:2:1 ratio of the homozygous dominant to the heterozygous to the homozygous recessive. But we can also think of this in terms of allele frequencies. And once again, this isn't to say that they only have 4 individuals. They could once again have 100 offspring. It just means that of those 100 offspring, we would expect 25 of them to be the homozygous dominant. We would expect 25 of them to be the homozygous recessive. And we would expect 50 of them to be the heterozygote, which is what you would expect from this ratio. But we also need to think in terms of allele frequencies. So rather than think of 100 individuals, it's just easier to think of 4 individuals. We have 4 individuals. Each of these individuals is diploid, which means that we have 1, 2, 3, 4, 5, 6, 7, 8 alleles. 4 of those alleles are the big A allele. 4 of those alleles are the little a allele. So we still have half the population. Half of the alleles in the population are the dominant allele. Half of the alleles in the population are the recessive allele. Now, it was pretty easy up to this point If we want to know what the allele frequency is in the next generation, or the genotype frequency for that matter in the next generation, how can we do this? Any ideas? Yeah, Sonia. Okay, how would you do that? So if we did it in a Punnett square way, how would we do it? With these, we have these three genotypes. Half the population is made up of this genotype. 1/4 of the population is this genotype. 1/4 of the population is that genotype. Yeah.
You just do like a Punnett square, except it's going to have like 4 across the top, and it's like 8 waves. Or it's like the video,
No, because you have to deal with each genotype separately. So what are the kinds of combinations that we could have? What are the types of matings we could have in this population? Let's say this is a population of 1,000 individuals. 250 of them are this genotype. 500 of them are this genotype. 250 of them are this genotype. How would we figure out what the allele frequencies in the next generation would be? Yep, go ahead. Okay, so we could take a homozygous dominant versus a homozygous dominant. They would produce nothing but homozygous dominants, but we would have to weight that probability of getting that cross by how many homozygous dominants are in the population. Then we could do homozygous dominants by heterozygotes. We'd give you this by that. But once again, we would have to weight that outcome by the weight of the heterozygotes combined with the weight of the homozygous dominants. We could also do AA by little a, then we could do little a by little a big A, etc., etc. Isn't that a pain in the keister? So there's a better way. We know that we can substitute the frequency of the big A allele and the frequency of the little a allele into a Punnett square. And when we do that, this doesn't become a genotype as much as it becomes this frequency multiplied by that frequency. This doesn't become a heterozygote, it becomes this frequency multiplied times that frequency. This is the same because it's still P times Q, and this becomes Q times Q, which is Q squared. The allele frequency that we've been dealing with all along has been a half, so a half times a half is 1/4. A half times a half is 1/4, 1/4, and 1/4. So we get 1/4 of the population will be the big A, the homozygous dominant. 2/4, 1/4 plus 1/4 is the heterozygote, and 1/4 is the homozygous recessive. We get the same genotypic ratio that we got when we just worked through the Punnett square, but we didn't have to do the Punnett square. All we had to do was a little bit of math. So this leads to this equation, P squared plus 2PQ plus Q squared equals 1. That equals 100% of the population. Q squared is the genotype frequency of the homozygous dominant. 2PQ is the genotype frequency of the heterozygote, and Q squared is the genotype frequency of the homozygous recessive. Now, if we want to know what the genotype frequency is in the next generation, all we need is the allele frequencies, P and Q, of the previous generation. And we can plug it in, but what we're going to get when we plug it in is we're going to get the same thing that we had before. 50% of the population of alleles is the big A allele. 50% of the alleles in the population are the little a allele. And we could do it generation after generation after generation after generation, and P and Q will stay the same. Why? But what if there was selection against this genotype? That genotype would become less frequent in the population, and over time, the allele frequency would change because you would have fewer recessive alleles because there was some selection acting against individuals with this genotype. So it doesn't have to stay the same, it just does in this case. If you introduce selection into the process, then all of a sudden you have allele frequencies changing. So that's one way in which it doesn't have to stay the same. What are some other ways in which it doesn't have to stay the same that you can think of? Just imagine. Yeah.
Maybe if there was like an extinction event of some kind.
Well, I would put that under the category of selection, unless, because if it kills off everybody, then the allele frequency falls to zero because there's nobody around. So you would have to think less severe than extinction, in which case it would just fall under the category of probably some form of selection acting. Think about other things. Yeah?
Have a genetic mutation.
Okay, so you could have mutation. So you had allele A, dominant allele, recessive allele A. You could now have a new dominant A prime come in. And now all of a sudden, it's really rare, so it's not changing the allele frequency much, but it's no longer 50-50. It's, let's say that you have 100 individuals, it's now 49, 49, and 1 out of 100. Oh yeah, sorry. 2 out of 100. So yeah, mutation is certainly one way that you can get changes in allele frequency. What's another way you can get changes in allele frequency? Oh, I didn't bring my coins. Oh, I did bring my coins today. Hold on to that for a minute. How many individuals in this population? 8 individuals. They're all diploids, so how many alleles do we have? 16 alleles. How many of these alleles are the dominant allele? 8. So P is 1/2, Q is 1/2. Heads you live, tails you die. Flip it. Oh, flip it in a random way. Don't you know how to flip a coin?
Not really.
Put it on your finger with your thumb, flip it so it flips over a lot, and then catch it and tell me what it is. Heads, live, yay! What is it?
That's tails.
Tails, oh, sadness. Continue. What's weird about it? It's like an American quarter.
Heads.
Heads, yay!
Tails.
Tails, sadness.
No tails.
Tails, sadness.
Tails.
Sadness, man.
Heads. Yay.
Yay!
Tails.
Tails. Yay! Oh, no, tails. Sadness. Okay, so now we have three individuals left. How many alleles do we have? 3, 6 alleles. How many of the big A allele do we have? Allele frequency has changed. Where's my quarter? I need a gumball later. There was death. Was death related to genotype? No, it was random. Random according to a terribly flipped coin, but still random. It was just random. It was the flip of a coin. It didn't have to do with whether you were heterozygous or homozygous recessive or homozygous dominant. It was random. This is a process that we refer to as random genetic drift. We could let these individuals breed, look at the next generation, and flip the coin on those individuals, and it might go back the other direction, towards more of the recessive allele being present than the dominant allele. So it's called drift because allele frequency just kind of wanders around. It goes up, it goes down, it goes up, it goes down. But it's not due to selection because the pattern of mortality isn't related to your genotype. Selection is when the pattern of mortality is related to your genotype. Homozygous recessive individuals survive less well than homozygous dominant individuals. So let's review for a minute. We've talked about 3 different things. Selection can change allele frequencies. Mutation can change allele frequencies. Random genetic drift can change allele frequencies. There's one other way in which we usually think of random genetic drift, at least in these beginning stages. One more way in which we think of allele frequencies changing. There are actually about 8 of them, but at this stage we're going to teach you 4 of them.
Fourth one is the most dramatic of it all. It can be
Yes, ma'am. Just do it. Bottleneck effect. Yeah. So that's a special case. That's a special case of this thing that I'm trying to get at. Small population size. Actually, we're going to teach you 5 of them. Small population size. Let's think about the bottleneck effect as a special case of small population size. Well, how do I want to do this, Nathan? Yeah, so small population size is actually just a special case of genetic drift. Genetic drift is more prominent in small populations. So imagine rather than a population of 8 individuals, we had a population of 1,000 individuals. So I'm not going to write 1,000, but we would have 250 of this genotype. We would have 500 of this genotype. We would have 250 of this genotype. If we went through and flipped a coin, even a poorly flipped coin, I'm going to give you shit about this the rest of the semester, just so you know. If we flip a coin, small little fluctuations due to the random nature of it, they're not going to have as big of an effect on the overall allele frequency as they would if you only have 8 individuals. Because you only have 8 individuals, you only have 16 alleles. So if you lose one genotype, especially if it's a homozygous dominant or homozygous recessive, all of a sudden you've lost a large chunk of your population of alleles, and that has big effects on allele frequency. So random genetic drift is most pronounced in populations that are small. And so if your population is infinite, random genetic drift goes away.
You flip that coin, how happy I flip.
you get the heterozygous, about a quarter of your to get the homozygous dominance, about a quarter of your flipping rate, or so dominant 500 flips, so you can homozygous recessive.
Yeah, and you can simulate this on a computer, and it works out really nicely. But I'm thinking of something else. When we talked about the coots being ornamented, the baby coots are ornamented because the parents choose the coots with the orange feathers more than they choose coots that have less orange on them. Can you think of other bird species that are ornamented? Okay, cardinals are ornamented kind of in that they're bright red. I'm actually thinking of something even more extreme than that.
Peacock?
Peacock. What's the special thing about peacocks? What's that? They don't fly. What else is special about them? They have these huge long tail feathers. Why do peacocks have huge long tail feathers? Oh, yeah. It's sexy. Because peahens, when they look at a male with a really long tail feather, they're like, oh, I want to mate with that dude. So peahens prefer males with long tails, but there's also variation in choice in peahens. Some females are choosier than other females. And so these two traits, the choosiness of the female and the elaborate trait of the male, become genetically coupled with one another, such that if you have the dominant gene for long tail feathers, chances are peahens that are choosy will also be carrying that same gene that is expressed, and chances are that male will also be carrying that choice gene. They become genetically coupled over time, such that in this case, only homozygous dominant individuals breed with other homozygous dominant individuals. They don't breed with heterozygous or homozygous recessive individuals. In this way, you get the concentration of that particular genotype in the population because like genotypes are always breeding with like genotypes. And so one of the other things that can happen is when we have what's called assortative mating. When who you mate with is determined by their genotype. It's a form of selection. It's sexual selection in this case rather than natural selection. So what we've just done is we figured out 4 of the variety of things that cause allele frequencies to change. Selection will tend to make allele frequencies change. Mutations make allele frequencies change. Genetic drift acting in small populations make allele frequencies change. And non-random mating make allele frequencies change. If you're in a population and there's no selection, there's no mutation, your population is infinitely large such that random genetic drift doesn't have an effect, and there's no assortative mating, all the mating is just random between genotypes, allele frequencies will not change. Let me repeat that. There's no selection, no mutation, no genetic drift because your population is infinitely large, and no assortative mating. Allele frequencies will stay the same over time. What's the term we use for when you're at a state where something is staying the same in science? Yeah. Somebody said it. Conway, equilibrium. So we could say that allele frequencies will be at equilibrium if these things aren't occurring.
You mean genotype frequencies will be in equilibrium. Your allele frequencies could be anything between 0 and 1 as long as it's something.
but they'll still stay the same.
Yeah.
Yeah. They could be 0.7 and 0.3, but they won't deviate from that. And if you have 0.7 and 0.3, then the big A allele would be 0.49. The little a allele would be, the genotype would be 0.49. This would be 2 times 0.7 times 0.3, which would be 0.41. And this genotype would be 0.09. So 0.09. So that would be the genotype frequencies, but the allele frequencies of 0.7 and 0.3 wouldn't change. That would just be what you start with, and that would be where they stay as long as those things aren't happening. We get these genotype frequencies from this equation. This was an equation derived by 2 guys named Hardy and Weinberg. And so this is called the Hardy-Weinberg Equilibrium equation, because if you run allele frequencies through this equation, it will give you the genotype frequencies, but the allele frequencies will not change from generation to generation as long as there's no selection, no mutation, you have infinite population sizes, and everybody's mating randomly. Stay the same. Generation after boring generation after boring generation. And that's what we see, and this is in the PowerPoint, which now I've set down my clicker somewhere. So this also, the PowerPoint for when you get home and are doing this goes through this all. This is where the Hardy-Weinberg Equilibrium comes from, and this is where you get the genotype frequencies from it. Any questions about that? All right, how many of you have ever seen a Manx cat in real life? Oh yeah, you have. Oh, Dr. Judd and I are the only ones. Manx cats, I feel like that's every year. Manx cats are these cats. They have no tail. And they have short, stocky bodies, kind of short legs. Everything's kind of compressed. Well, Manx cats are all heterozygous. They have a dominant allele and a recessive allele for this particular genotype. And normal cats are homozygous recessive. So homozygous dominant individuals are spontaneously aborted before they are given birth to. And so this was first reported on the Isle of Man, which is an island off the coast of England, I believe.
Yep.
You guys follow professional cycling at all? Mark Cavendish, the greatest sprinter of all time. He is from the Isle of Man, and they call him the Manx Missile because when he gets close to this finish line, he just shoots towards this finish line and blows everybody away. He's also now won more stages of the Tour de France than anyone else in history because he is a good sprinter. He's a good sprinter. If he can get to the finish line in that front pack of individuals, he sprints to the finish line and crushes everyone. He's the Manx Missile because he is from the Isle of Man. That's why these are called Manx cats. So, homozygous dominant individuals die before they're born. They get conceived, but they die before they're born. So, all the cats that you see out in the world that are Manx cats are heterozygotes. Normal cats are homozygous recessive. So let's imagine that we're on the Isle of Man and we have a population of Manx cats. Actually, we have two Manx cats. Let's just think of it simple as 2 Manx cats. They have this genotype. We're going to cross them. How many alleles do we have? We have 4 alleles because we have 2 diploid individuals. What's the allele frequency of the dominant allele? Allele frequency of the recessive allele is 1/2. We can do this with a simple Punnett square. This individual can make these gametes. This individual makes these gametes. And we get homozygous dominant, 2 heterozygotes, homozygous recessive. It's your standard Punnett square with 1 trait. What's the allele frequency at this stage? You have 4 combinations here. 2 of them are the same, but what we're really concerned about is allele frequency. What is P and Q? Well, at this point, it's 1/2 and 1/2. 8 alleles, 1, 2, 3, 4 of them are the dominant allele, 4 of them are the recessive allele. But of course, somebody's jumping ahead. What happens to that individual there? This individual dies. So do we have 8 alleles now in the population? No, we have 6 alleles. 2 of those alleles are the dominant allele, and 4 of those alleles are the recessive allele. So 1/3, I heard this, somebody say this, this is 1/3 and 2/3. So selection on this population has gone from the parental generation to the F1 generation, where P was 1/2 to now P is 1/3. That should be a 3. Frequency has gone down. If we want to know what the population is like in the next generation, what do we have to do? We could do a crossword, we could just do what? Hardy-Weinberg Equilibrium. And this is why you're going to see that fractions help. P squared plus 2PQ plus Q squared equals 1. P is 1/3 squared plus 2 times 1/3 times 2/3 plus 2/3 squared. 1/9, 4/9, and 4/9 should equal 9/9. This is the frequency of the homozygous dominant, frequency of the heterozygous, and frequency of the homozygous recessive. But of course, what happens? 1/9 dies. That's shown in the animation. 1/9 dies. So now do we have 9 parts of our population? No, we have 8 parts of our population. This is 8/8, which means this is 4/8, and that is 4/8. So half of the genotypes, not half of the alleles, half of the genotypes are heterozygous, half of the genotypes are homozygous recessive. That's kind of saying that we actually have half this and half that, so we can think of this as being a population made up of 2 individuals. If this is really just 2 individuals, how many alleles do we have total? 4. How many of them are the dominant allele? P is 1/4. Q is 3/4. In the F2 generation, P is now 1/4. It's gone from 1/2 to 1/3 to 1/4. If we want to go, so this is also animated in the PowerPoint for you. Blah, 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 blah. Want, 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 want. What do we do next to get the next generation? Just apply Hardy-Weinberg again. 1/4 squared plus 2 times 1/4 times 3/4 plus 3/4 squared. So 1/16 plus 6/16 plus 9/16, 9, 10, 16. That's 16/16. But what happens? 1/16 dies. So this isn't really 16, this is 15/15. So this is 9/15, and this is 6/15. It's a common divisor of all of these. 3. If we divide this by 3, that's 2/5, and that's 3/5 to make 5/5. You can think of this as 2 individuals in a population of 5 individuals that are this genotype, and 3 individuals in a population that are this genotype. How many alleles do we have total? We have 2, 4, 6, 8, 10. We have 10 alleles total. How many of those are the dominant allele? 2. 2/10, which means 8/10 are the other one, which is 1/5, 2/5. The third generation, P has now fallen to 1/5. Do I need to continue belaboring this point? Because I can. I'm a one-trick pony. I can do this all day long. I've never done it this far, but I can. 1/5 squared, 2 times 1/5 times 4/5, 4/5 squared, 1 25th, 8 25ths, and 16 25ths. 16 and 9 is 25 25ths. What happens? 1 25th dies, so we really have 24, right? 24, 24ths, 16 24ths, 8 24ths. Common divisor here is what? 4. This is then 2 6ths, and this is 4 6ths. This is the homozygous dominant, heterozygous homozygous recessives. You could work it out just the same way that we've been doing. Much harder to see this pattern if you're doing this with decimals, which is why I do it with fractions. Also, the math is just quicker for me doing it by fractions. Over time, what do you think is going to happen to the big A allele and the little a allele? Yeah, this becomes less and less frequent. And this becomes more and more common. Until what happens? You might expect the little a to just finally go away, right? Because it's becoming less, the little m, the recessive allele will become less and less common because the dominant allele gets hit every year. Because it always produces some homozygous dominant individuals. Will it go totally away? Or will there always be some dominant alleles hanging out in the population? Okay, there will always be a little bit of dominant individuals in the population. Why? But why is the frequency never going to go to 0? It's always getting passed down because what do you always have? In what genotype? The heterozygous. So there's always heterozygous. They look funny because they're Manx cats, but they don't die because of it. And so you always will have this dominant allele hanging out in the population because it's harbored in that heterozygous portion of the population. So this is what it looks like if you run a computer simulation on it. Half, third, quarter, fifth, 1/6, etc., and it gets more and more and more uncommon, but even after 100 generations, that dominant allele is still hanging around, and that dominant allele is still hanging around because heterozygotes still live. Yeah, question?
So out of the things that you listed on the board, which of those does this classify?
This would be selection because the fact that you die in childbirth is totally tied to your genotype.
Natural selection?
So this would be a form of natural selection, yep. So what distinguishes selection from the other things is that your ability to survive or your ability to reproduce is related to the type of genotype that you possess. Random genetic drift is not that way. You could be walking down the street and a piano falls on you and you're dead. You just happen to be in the wrong place at the wrong time. Has nothing to do with what your physiological capabilities or anything else that's governed by genetics. You just really happen to be in the wrong place at the wrong time. When the cable snapped, you couldn't hear it coming, and it just fell on you, crushed you to death, and now you're gone. You could have all the superior genes in the world, and the piano still would have dropped on top of you.
At the beginning, Dr. Berkeley said that one way to think about evolution is changes in gene frequencies over time. That list is a series of different ways that gene frequencies change over time. That's why when we talked about evolution in the first unit, I said now the question is the primary method of adaptive evolution. There are other ways you can change the look frequencies, and we just listed them. So that's the only and the primary method.
Now, if you're walking down a sidewalk in New York and people are moving a piano up into a penthouse and the cable breaks and it falls down and crushes you, and there's no way that you could have seen it coming, it just literally falls out of the sky, crushes you and kills you, that would be kind of like random genetic drift. Now, if you live in an environment where pianos fall out of the sky on a regular basis, you might see that population evolving, better hearing, eyes on the top of your head, sprint speed to get out from underneath the falling piano, in which case your survival is tied to genes that you possess that allow you to detect falling pianos, get out of the way of falling pianos, etc., etc. That's no longer a random thing. That's now a feature of the environment. That environment then exerts selection on individuals, and depending on what your genotype is, you're either better equipped to get out of the way of falling pianos or you're not better equipped to get out of the way of falling pianos. Difference between random genetic drift and natural selection. Okay? And these are actually what we sometimes present to you as the four forces of evolutionary change. Selection, mutation, random genetic drift. Actually, there's another one. This is actually a form of genetic change because, remember, selection can either be natural selection or it can be sexual selection. And random genetic drift is related to population size. There's this other force that we haven't talked about, which is migration, which we didn't mention with Hardy-Weinberg Equilibrium, but it's another one of the assumptions of Hardy-Weinberg Equilibrium. If I pick up my blonde hair, blue-eyed genes, trust me, my hair is blonde. You can't see it, but it's blonde. It used to be blonde, now it's gray probably. When I was younger, my hair was blonde and I have blue eyes. When I was a postdoc, I got a job in Puerto Rico, and Puerto Rico is an island that has very few blonde-haired, blue-eyed people. And so I took my blonde-haired, blue-eyed genes from central Pennsylvania at Shippensburg University, where I was teaching for a year, and I took them to Puerto Rico, and all of a sudden Puerto Rico has one more set of copies of blonde-haired, blue-eyed genes, and Pennsylvania lost those blonde-haired, blue-eyed genes. So just me moving changed the allele frequency of both the population I was leaving and the population that I was going to. So migration also affects this. These are the four forces of evolutionary change. Selection is either natural selection or sexual selection, non-random mating. Mutation brings new variants in. Random genetic drift causes changes in allele frequency because of random death. But then one of the other ways that you can get allele frequencies changing is through moving alleles from one population to another population as individuals migrate. And this now seems like a good time to define what evolution is. In its purest, simplest form, evolution is just a change in allele frequencies over time. You can define it other ways, but this is a way that probably applies to all the ways that I can think of as evolution happening. Can you think of a situation that that doesn't apply to?
No, my only critique with this part is that we don't ever, when we talk about Hardy-Weinberg Equilibrium, we're always talking about change in the whale frequency of a plant in the context of a single population of species. But we never touch on, and we rarely touch on in the context of Hardy-Weinberg Equilibrium, is the interaction between this kind of change and the formation of new species.
Yes. So, but the formation of new species, if you let this happen long enough, you have the potential to form new species. And so we oftentimes refer to microevolution and macroevolution. Microevolution, evolution that occurs along a single lineage, such that that species changes over time. So species A, if this is a scale of some morphological trait, let's say that it's beak size from small beaks to large beaks. Species A may start off here, and natural selection may push species A towards larger and larger beaks until at some time in the future, species A is still species A. They just all have larger beaks. This is what we would refer to as microevolution. But if something happens such that seed supply is really low, they eat it all up, they start competing amongst themselves, and some individuals who have small beaks eat one section of the seed supply. Individuals that have large beaks eat another section of the seed supply, such that over time, one part of this population goes after small seeds, another population goes after big seeds. You end up with these two morphologically distinct groups that at some point in here we begin to start calling species B and species C.
If they start mating preferentially rather than one.
Yes.
Otherwise, you can just have morphs from one species.
Yeah. And this is the situation that's going on in Hendry. They're in the process of doing this diverging. What makes it possible for them to come back together is that the whole time that they were diverging from one another, members from this lineage would breed with members of this lineage, members of this lineage would breed with this lineage, and even when they were most distinctive, we still had those two lineages interbreeding with one another. So that as soon as the selection left that was driving them apart, that selection is no longer there. The fact that they're interbreeding allows them to converge again morphologically because there's no selection keeping them apart. And they're free to interbreed, and when they interbreed, they produce intermediate-sized finches, and those intermediate-sized finches survive rather than die. Is that kind of what you were getting at, Nathan?
I'm curious if anyone has any questions about this. That was in my mind, that's not what I was thinking of, because in plants you can have speciation that doesn't take this long time of separating, and then isolating it. You can sometimes get reproductive isolation.
Very quickly. It happens way more often in plants, but it still happens occasionally in animals.
And when it happens, you have that sudden change gives you a full population, so genetic drift still plays a role. So I would still agree with that's why I had that long pause when you asked me if I agree with that.
Yeah, so you're thinking of ploidy events? Yeah. So one of the things that plants do that animals do less often is they can duplicate their genomes and still function. And so you might go from a species that has a diploid genome and now it has a tetraploid genome or a triploid genome. And plants, for whatever reason, handled that better than animals do. And I don't know the reason for that.
I don't know why that animals can't, but even if you double your chromosome, they still just have to pair up and do mitosis and meiosis. So.
It's just really rare. There's a frog species that occurs here in Missouri, the gray tree frog. It's actually two different species. There's Cope's gray tree frog, and then there's the gray tree frog. And one of them is diploid, and one of them is tetraploid. And the only way that you can tell the difference between them is to look at their blood cells. Their blood cells are actually different sizes because the nucleus is in the blood cell in amphibians. And so the tetraploid actually has bigger blood cells than the diploid. You can also listen to their call. They call at different rates, so you can distinguish them by their call. or you could just sequence them, but just listening to them calling in a pond is far easier than sequencing them. You could do a karyotype, I guess, analysis of them, but once again, it's easier to just listen to them calling.
The picture I showed you with all the little stripy chromosomes, that's what he means when he says karyotype.
Yeah. It's by far the most complete presentation we've ever had of this material, I think.
Yeah, as long as you get those four, remember that you talk about evolution in this context. You're usually just thinking about this kind of change. And we will later be certain about it.
Yep. Yeah, that's a definition that applies to both of these things. If you're not having changes in allele frequency over time, that macroevolution isn't happening either. So now that we've kind of talked about populations that stay the same over time because there is no selection, there is no mutation, there is no small population size, there is no migration, things like this, we can go back to thinking about these additive genetic traits that we talked about because the Darwin's finches, their beak size is presumably an additive genetic trait. And so we need to think about these in productive ways. When we think about selection in an additive genetic trait, we don't have to actually think about the alleles. We don't have to think about the genotypes. We can actually think about this as a trait distribution, and we can ask ourselves, well, what happens to this trait distribution if we select against, for example, small-beaked birds? And what we would expect to happen with this population is what in future generations? If this is the distribution that you begin with, and you select against small-beaked finches, what happens? Or short individuals. If we're talking about height. You get more tall individuals. So selection is going to push you to a population that has more dominant alleles in it, and so you get taller individuals. What else is going to happen to this distribution? So it's going to shift to the right, but what else will happen to it? I'm not sure what increase you're talking about.
Like when it sheds, so it's gonna
Oh, yeah. You're looking ahead, aren't you?
yeah.
You're looking ahead. Is this the steepness that you're talking about? So what you're trying to say, and not doing a very good job of it because we haven't given you the language to say it yet, is it's not only shifted, and this should not be S, this should be R. R is the evolutionary response to selection. I need to change this slide. The slide's been wrong for a long time, and I just never go back and fix it. You can designate this as R as the evolutionary response to selection, which is why it's R. Another way that people define this is delta Z, which is the change in the average trait value. So what happens when selection acts? Selection is acting on this population, this portion of the population. That pushes the population to the right, but it also makes it less variable. Notice how this is a wide amount of variation in the pre-selection population, and it's a narrower amount of variation in the post-selection population. So what you were trying to get at is that the post-selection population is less variable than the pre-selection population was. And that does make this side of this curve steeper, but you were trying to get at the squeezing of this population in terms of its morphology. This is what happens with directional selection. Selection occurs against one part of the distribution or the other part of the distribution, and it pushes it in one direction. That's why it's called directional selection. Stabilizing selection is when you have selection against both extremes of the distribution. Short individuals are not favored. Tall individuals are not favored. It's average individuals that are favored. What do you expect to happen to the average phenotype and the variability to happen? What do you expect to change in terms of the average trait value and the variability in the trait under this scenario? You have selection against the extremes. Yeah. Okay, the average becomes more frequent, so has the average trait value changed from one generation to the next? No, it stays the same, but what happens to the variation? It goes down. That is exactly what happens. So in this case, the average trait value is the same in the pre-selection and post-selection scenario, but the post-selection population is less variable than the pre-selection population. So in this case, R would be 0, as opposed to R being positive or R being negative. In disruptive selection, average individuals are selected against. What do you expect to happen to the average trait value and the variability under this scenario? This would call us bimodality. So would the average value change? Taking it exactly out of the middle. So you have an equal distribution on either side. So you wouldn't, Trevor? Yeah, so the average wouldn't change, but what would happen to the variability? It would increase. Because you have more of the trait distribution distributed out here and more of it distributed out here, so the values, if you remember how variance is calculated, it's the difference between the individual trait values from the mean squared. So in this situation, the pre-selection population, a lot of the individual trait values are very close to the mean. Once you take selection against the average individuals, you spread this population out. Now more of the individual values are further from the mean, so the variation goes up. So this statistical concept of variance is useful in this context.
And this is a good reason why you want to think about how long your fat distribution is, and not how steep the sides are.
Yeah. Mm-hmm. So those are three types of selection that can occur. Directional selection moves the average value of the trait either up or down, and it reduces variation. So the mean changes, but it changes directionally, and the variation goes down. Stabilizing selection, the mean doesn't change over time. You just get a trait that is less and less variable over time. Disruptive selection, the average value doesn't change over time, but it becomes more and more variable. Which of these is going on with Darwin's finches in the paper that you are studying? Okay, be careful here. Where is disruptive selection occurring? Where and when, actually? Okay, so in that case, we have stabilizing selection, but when and where is stabilizing selection happening? Academy Bay when? Recent Academy Bay, we have selection against big and small big birds, and the intermediate sized birds are becoming more common. Where and when do we have disruptive selection happening? Forget Barrow Bay, it's a weird anomaly. El Garapatero now and Old Academy Bay. That was an example of disruptive selection. So in that paper, they talk about two different forms of selection. They don't talk about it in these terms, but now you have the vocabulary to talk about it yourself. We have disruptive selection going on in the 1960s at Academy Bay. Now in Academy Bay, we have stabilizing selection happening, but disruptive selection is still going on in the present day at El Garapatero. Questions? All right. So these types of traits that have continuous variation underlying them, we can study their heritability mathematically or statistically rather than doing Punnett squares. And so when we talk about a trait being heritable, we're talking about the degree to which the phenotype in parents does a good job of describing what the phenotype will be in offspring. So heritability is a measure of family resemblance. But it's family resemblance that is undergirded, can't spell. Undergirded by additive genes. So when we want to know how a population is going to respond to selection, what we do is we look at the phenotype of the parents and we look at the phenotype of the offspring and we run a linear regression between these two traits. This is commonly referred to as a parent-offspring regression. And it's the slope that matters. So in this figure, we have the average parent height because there's only two parents. It's the mid-parents, the midpoint between the two parents. And we have the average of all of the offspring. This is actually a data set from a group of students in evolutionary ecology a number of years ago, where we actually had 64 students in the class, which was crazy. Don't ever want to do that again. This is the equation. Average offspring height is determined by the y-intercept plus the slope multiplied times the mid-parent height. So it's the slope value that tells you how heritable a trait is. A trait can go from a heritability of 0 all the way up to a heritability of 1. And those are the theoretical minimum and maximums for heritability. And so heritability for height in this particular sample of human beings was actually quite high. But now we can think about selection acting on this height. Imagine that we are Nazis and we want to breed the Aryan super race and we want that race to be tall. We could basically prevent everybody below the average height from reproducing. That's why they are in red, they are dead. This would be the average of the population before selection occurred, and this would be what we would expect the height of humans to be before selection occurred. But this mean changes after selection because these individuals here are not included in the parental population. So the parental mean changes. When the parental mean changes, what do you expect to happen to the offspring mean? It's also going to change. And the degree to which it changes is determined by the slope of this line, which is why the slope is the heritability, not the y-intercept or anything else. So this shift in the parents is selection. The change in the offspring population is delta Z or R, the evolutionary response to selection, and the degree to which that number changes from the before selection value to the after selection value is determined by the slope of this line. Questions about that? We're going to explore this further on Monday. Did you look at the topic for Monday?
It's density times population growth, and I
Okay.
think that's population homework.
All right. There are slides in the remainder of this presentation. I especially want you to look at the next slide between now and Monday, because that'll help us get through that material a little better, and it will keep us from taking up too much of Dr. Judd's time to talk to you about population growth. All right, enjoy your weekend, and I will see you on Monday.
Homework. You just finish it up and then
Is there